the tail end of 16 on the next exam. So you will see a question about the reducing sugars versus oxidizing and all that fun stuff. So when we're looking at reducing sugars, it's that really weird definition because a reducing sugar is one that in the course of its reaction is oxidized. Okay? And that's because it is reducing something else. So what you are looking for in that process is an aldehyde, because aldehydes will actually react or be reduced. And ultimately, you need to be looking for that acetal. So we have that acetal functional group right there. I don't mean ketal, I mean acetal. Okay, so we are looking at a distinct difference here. Okay, so the acetal really only shows up. It's a way to hide the fact that we have an aldehyde. Because what happens if we break open that ring structure? We end up with an aldehyde. Okay? That's what's getting reduced, is ultimately that aldehyde formation. If we had a ketone or a ketal, it's not going to be reduced. Okay? And ultimately, it needs to be even further a acetal. Okay? We need to have access to that OH. That's of critical importance here. As soon as we push it all the way to the acetal form, it can't actually be oxidized, and it would be known as a non-reducing sugar as opposed to a reducing sugar. And that typically happens when we go through and look at our disaccharides or our polysaccharides, because what will end up happening is we can form linkages between our anomeric alcohol to another carbon structure. That now bonds up that position, so it can't be reduced anymore. Okay, and we'll look at those in just a second here too. And that's really what's happening when we move up to the disaccharides and polysaccharides, is that we form a glycosidic bond. So that glycosidic bond is just in reference to putting a carbon on our anomeric oxygen. So there's our anomeric oxygen. If I bond that out to any carbon group, I now have a glycosidic bond. Okay, and that glycosidic bond now shuts down the reducing ability, okay, or oxidizing ability, depending on what you're referencing. Okay. Yeah, it's just what it is. So we can get some common disaccharides. So we got a couple options here. We're taking alpha-D glucose and we're linking them in slightly different fashions. Okay, because we're taking the same structures and we're now bonding them in slightly different fashions, we have to come up with some way to name them. And so that naming system, you should vaguely remember from the sapling homework and theory. What we're trying to reference is, where is that linkage? Where's our bond actually forming? So we get a glycosidic bond going that way, and we get one coming back the other direction. Okay, so within that naming system, we need to name each of those positions. So we'll start with our leftmost structure. And what we're saying by that name is that we have a bond between carbon number 1 and carbon number 4. You'll notice that the alpha is showing up in there. That alpha is in reference to the conformation of our anomeric carbon. Okay, so if we take a look at our anomeric carbon, that is carbon number 1. So there's our linkage. If we move to the other side, and we start our numbering from our anomeric carbon, that's over here, 3, 4. So we are forming a glycosidic bond between carbon 1 and carbon 4. Okay. To add information to that, because the anomeric carbon is involved in that glycosidic linkage, I need to specify where that OH is aimed, its relative location. Is it in the up or cis relationship to our CH2OH, or is it trans? Okay. Because it's still carbon number 1, but where that OH is located relative to that carbon number 1 is going to be different. So we have to add that information into this. And we add that by including our alpha or beta in front. Okay. So in this case, we take a look at our oxygen in our anomeric position, and we notice that it is underneath the ring, which is trans to the CH2OH, which should make it an alpha glucose. So we now have alpha 1,4. because we're referencing the bond between the alpha 1 position, or the 1 position, which is alpha configuration, and the 4 position of our second glucose structure. Okay. If we move to the next one, now what's our relationship? Okay. We still have a bond at carbon number 1. I don't know why I'm numbering all of that one all the way out, but I am. And the other one, we again start our numbering at the anomeric carbon of the other structure. Where's our glycosidic bond linked? Between carbon 1 and 6. So we're now referencing the 1,6 position. Is our anomeric carbon involved, or our anomeric position involved in that glycosidic linkage? If we say 1, then yes, it absolutely is involved. So I now need to specify what its relationship is. Okay? Is it alpha or beta? So we go back, our O, and this is where it gets weird, they show this stupid curved bond. Okay? That curved bond is trying to say that it is aimed downwards, but we've now curled it up so that we can maintain the Hayworth view for both of these structures. Okay, and not stagger it so it looks weird. Okay, but that's all they're trying to show by that conformation. So our oxygen is actually down, our CH2OH is up. We still have a trans relationship. The trans relationship was alpha. So again, alpha 1, 6. Okay, kind of makes sense? So now we're just into factual stuff. You got a bunch of common disaccharides. Definitely in the last time I taught this, I had students memorize at least one of these and then asked what those structures were. I don't see the point, quite frankly. But you've got sucrose, lactose, maltose. They're combinations of your different carbohydrates, and they're our most common ones. And there are certain conformations in which they've been put together. Okay? So you may be asked something along these lines in, say, a homework or maybe on a future pre-health test if they were really trying to push at that information. So you should at least review them, see what units or what sugars are being involved in each of those and what types of linkages they have, because you could be held responsible for that in a future class. Okay? After that, we move up into the polysaccharides, and with our polysaccharides, we can now get some either structural features coming out of it, or we can get just raw energy storage. So one of the more common polysaccharides is cellulose, and it's a massive structural component to plants. Okay? So all they're really talking about is now what's the linkage between all of those, and ultimately it's kind of interesting because what sugar are we looking at using to make cellulose? Yeah? Glucose. But glucose is something that we use, so why can't we process cellulose? Okay, and that's because the types of linkages used in cellulose don't facilitate or we don't have enzymes that are designed to process that. Okay. So what does that mean? Well, that means cellulose doesn't get processed, which means cellulose can be used as structure. Okay. Or fibers. It's not going to really do much except hold structure together, which if we think about it, does that make sense? Do we want to be able to process our walls? It would be cool if we just reached up and just punched a hole through the wall, grabbed it, and walked off with it. Our buildings would start to fall apart. Okay, so it makes sense that cellulose is not easily processed because it is contributing to our structure. Okay? So you get lots of hydrogen bonding within it. In fact, all of your carbohydrates are going to have lots of hydrogen bonding. That's one of the reasons why they have a lot of energy storage within them, because of all those potential charges that can come out of it. That hydrogen bonding can also allow for a vast network of stronger interactions between chains of our polysaccharides, and that can improve structural features. Okay. The biggest thing to note here is the type of linkage we're getting at is a beta 1,4 glycosidic linkage. It's that beta conformation ultimately that makes it difficult for humans or most organisms to process. Okay. So it's that type of linkage, or it's that type of position about the anomeric carbon that really prevents any kind of processing. Okay. We can jump up to starches. Starches are processable. Sure, processable. Okay. One of our biggest conformational changes is that you'll notice that we're looking at, instead of beta conformations, we're looking at alpha conformations. That subtle switch in that geometry now changes how it is stored and processed, and we can now actually break that down. Okay? So there's a couple different types of starches. You can get amylose and amylopectin. Ultimately what these are just variations off of the same pattern. Okay? Your amylopectin has a lot more branching within it. Amylose has no branching. So if you did a polymer lab, in theory there were a couple different types you could do, and you could look at just straight chain polymers, or you could look at cross-linked polymers. Well, if we have a cross-linked polymer versus a straight chain, what ends up happening to the mobility of that polymer? Okay, for instance, we draw a bunch of circles. There's a polymer. What could it potentially do? Those are all. It can coil. All of those single bonds can freely rotate. As those single bonds rotate, we get a lot more motion. What happens if I now cross-link that polymer to another polymer chain? So cross-link just means ta-da. Okay? Or if I cross-link it a whole bunch, does it get as much mobility? No. So it's going to change its way it interacts with the other environment. It's going to tend to solidify and make it a lot more rigid. Okay? So we're going to get slightly different variations from that and different properties for why we would want to do that. One of the fun ones that you might see just on a pre-health test would be testing for starches. Okay, your book has kind of a nice picture there, though it looks a little bit odd. What they're referencing is that iodine molecules can saddle up in between a starch helix, and what that does is now generates a proper electron configuration that we get a color change. So we can test for the presence of starch, or test for the presence of iodine for that matter, by seeing that relationship. Okay, so it's one of the common tests actually for both starch and for iodine. Okay, if we're trying to look for starch, we could add iodine to it. If we see a color come out of it, hey, we've got starch. Okay? Or we can add starch to a reaction and say, is iodine present? If you see that color change, yeah, we got iodine present. So you can see those kind of subtle changes. It's kind of a neat little process. Might show up on some kind of pre-health test just because it's a big color change, and you could potentially see that in labs. As far as decomposing starches, you've got amylases. So there's a variety of them on what they end up actually doing and processing. Okay, so you've got beta amylase and alpha amylase. The alpha and beta are not in reference to alpha and beta anomeric positions. They're actually in reference to ultimately 1 and 2. Thank you, Biochem, for giving us nice, distinct naming systems. So your beta amylase is an exoglycosidase. So all that's saying is the exo correlation is what you're looking for. Your beta amylase will grab your sugar molecules or your starch molecules at the end of your starch molecule. So it's only on the exterior parts that your beta amylase will work. Your alpha amylase, on the other hand, is an endoglycosidase. So endo means that it can attack in the middle. Well, if it can attack in the middle, it can also attack on the outsides. So ultimately, your alpha amylase can completely process any part of your starch molecule. Right? Well, if we try and think about speeds, what is that going to mean for speed? If we have an enzyme that can only attack the outside versus an enzyme that can attack anything. Okay, alpha in theory works faster just because it can now react with any species or any part of our structure, whereas the beta can only work on the outsides. Okay, so it's more of a concentration issue than anything else. Neither of them can break your beta 1,4 glycosidic bonds, which happens to show up where? It's our cellulose connection. So neither of those amylases can deal with that. The other interesting aspect of this is that these will only really process within the polymer chain. But remember, if we move to amylopectin, we have our polymer chains cross-linked. Okay, so what these process is just within a polymer chain. They can't actually break apart two polymer chains away from each other. Okay, so what do we need? We need yet another enzyme to break those two chains away from each other, because those are now a different connection. Okay, that's where you're getting into debranching enzymes. Right, or at least that's the name that they're referred to as. Those will help break apart those extra glycosidic bonds. You'll notice you're looking at a slightly different location. Okay, and that's because you're looking at a different bond. We don't get starch in animals, but we get an equivalent. We can get glycogen. So glycogen is our equivalent to amylopectin. You're looking at cross-linking a bunch of sugar molecules. Lots of sugar, all sorts of fun stuff, very similar processes to starch. Okay? But because it's in an animal, we call it something else. Okay? Which makes it even more fun when we go through and try and talk about the enzymes that break it down. Okay? So what ends up happening with that? We get the absolute equivalent as our amylases, but they get called something else. Okay? Why? Because a different animal did it, so we have to call it something else. Kind of irritating, but that's just the way it's set up. Okay, so it's the equivalent of your beta amylase. So your amylases just break things apart, and that's really all you need out of it. Whoa, doubled that down. Other things that can pop up for your polysaccharides, chitin. It's the exoskeleton of your insects. These need to be much more rigid. So what we're going to start to see in these is more hydrogen bonding, stronger interactions between those polymer chains. Well, we talked really briefly about them, but if we take a look at your sugar molecules, it's not just a standard sugar molecule. Okay, you'll notice that what we see at the bottom of those, come on, work with me. Is an amide functional group on the bottom, not just a standard alcohol. Well, how is that going to change the intermolecular forces? Okay, we still have hydrogen bonding coming from our NH, but we now also have a hydrogen bond acceptor coming from our carbonyl. Okay, so we've got a lot stronger intermolecular forces coming out of these types of sugar molecules, so that when we then link them into these massive polymers, we're now going to get even stronger interactions between the polymers. That stronger interaction is going to lead to a more rigid, potentially harder structure. And that's what we end up getting when we shift to chitin. Okay? And I know I glossed over it before. You can look at the naming of it to kind of suggest that you're seeing a slight difference. If it was just glucose, you'd just see the glucose. But we've got a glucose molecule with an amine. There's the NH, and an acetyl group, which is the carbonyl CH3. Okay. If you really wanted to get into the nomenclature of it, I'm sure you could push that further, but ultimately all that's happening is chitin is a slight variation of it, and because we're looking at a more rigid structure, we need stronger intermolecular forces. Those stronger intermolecular forces are going to contribute by making variations off of our glucose molecule. Really the only thing I'd want you to get out of that. Cell walls, same kind of idea. We now go through and again vary our structure and the types of linkages, and when we go through and change our structures and change those linkages, we can get slightly different properties coming out of these. So again, change the sugar, yay, new carbohydrate, or a new polysaccharide with slightly new properties. So same idea as your cell walls. If we look at the sugars that are being generated in here, come on, pen. We have the same acetyl amino group that we saw on the other one, but you'll notice that we also have another acid functionality hanging off of that. That acid functionality is helping to contribute to stronger intermolecular forces. stronger intermolecular forces, we're going to start to get more rigid structures coming out of that. Okay? And that's where they come up with the different names, the NAM and the NAG. Okay? N-acetyl something.
Is that what an antibiotic breaks down? Is their cell wall?
I don't know that.
Usually breaks down
Bacteria in it,
peptidoglycan.
but I'm saying, doesn't it essentially break down the cell wall?
Peptidoglycan.
That's what I thought.
That's no, I can.
Oh, yeah, I know.
So I can take a look for you, see if I can find something on that, but I honestly don't know. It's going to have to target something. It's going to have to target something specific to bacteria. If you're trying to kill bacteria and not your own cells, you have to have some targeting mechanism.
Like, fluorine does the same thing in bacteria.
Well, it's going to be
Bacteria.
shifting different things, yeah, and targeting different molecules within them. So if this is something that only shows up within the cell walls of bacteria, you can target that by generating antibacterials that will then target those individual positions. Okay. The other option is you just use something that is relatively toxic, say like ethanol. It's a great antibacterial, because if you drown something in ethanol, it dies. Okay. So when you look at the antibacterial hand washes, almost all of them, the primary ingredient is alcohol. Okay. Because if you wash with alcohol, that ends up killing it. That's going to kill pretty much anything, but your hands get relatively free of that alcohol, and you don't end up dying or getting the harmful effects. But if we dropped you into a vat of ethanol, you'd have some problems. Yeah, that's not pleasant. That's awful.
Hand sanitizer. We had a guy crawled up with it one night. He kept like being in his room, and he like every time the nurses would go in, he'd be like curled up with like a bag of hand sanitizer. Because he would do this exactly the same thing.
Probably the same guy.
They'd sober him up, they'd like discharge him, and then he'd be back 20 minutes later, well like 30 minutes later, he'd be like drinking it outside. It's good.
Yeah.
Locked out.
It's probably the same guy.
He was drinking
So-
alcohol, the hand sanitizer, you get drunk?
Yeah.
Yeah, you can get drunk off it.
He can get it.
Wow,
out
I'd rather
of
do
the
Listerine if I was
within
that
5
broke.
seconds.
Listerine's cheaper.
Yeah.
Well, these are paying for it, it's just stealing from the hospital.
A lot that at the psych hospitals, we have non-alcoholic. You can get hand sanitizer that doesn't have the alcohol component.
I'm sure.
That's
I'm
way too
sure.
fancy for us.
Yeah,
Yeah,
some
it's
sort
probably
of,
more expensive, yeah.
you guys are more of like a constant liability for that.
So we have yet more types of polysaccharides that come out of this. This is an exciting chapter. So they list off a whole bunch of others, and you end up again changing the properties by changing the structures. Right? I'm kind of sick and tired of carbohydrates. The last one that I actually thought was kind of interesting was looking at the different blood types, A, B, O. What they're referencing is actually a carbohydrate tail that is hanging off of your particular blood cells. And that's what's giving you particular types, be it your A, B, or O. So what ends up happening is with that particular sugar sequence, that's what gets recognized as your own blood type. So if you start mixing bloods, you have antibodies that will respond to anything that is not that blood type. Right? So if you start mixing bloods, that's when you're going to run into problems, because then your immune system is then going to attack that foreign blood substance, because even though it's transporting oxygen, your body still recognizes it as something different. Okay? It's not part of your natural system. So that's why when we go through and do blood transfusions, you got to make sure that you've got the proper ones, because there are antibodies within your body that will then go through and attack anything that doesn't have that tag. Right? And those are ultimately carbohydrate chains coming off of that. So I thought that was kind of a neat parallel. Any questions about carbohydrates? Kind of glad to be done with just fact stuff for a while. So now we're going to move into chapter 15, and now what we've got is probably 5 minutes of loading here. So what we're now moving into is ultimately what I would consider to be the real aspect of biochemistry. It's all the metabolic pathways. And so what we see up on this slide is a pretty impressive image, which I did not include in the slides that I posted for you guys, because it takes the file size from maybe a few hundred kilobytes to several megabytes with just this single image. So what this image is actually showing you is all of the known metabolic pathways that we're aware of. Okay, so there are some metabolic pathways you are probably familiar with. Anybody got a guess? Krebs cycle. Where's the Krebs cycle? There's our Krebs cycle. So we will be talking about the Krebs cycle, the citric acid cycle, this semester. That is just a tiny little piece of this overall puzzle. Okay? Very, very impressive, the amount of information that comes out of this, which should kind of lend you to the idea of biochem has a whole lot of stuff going on within it. It's very hard to take a class and summarize everything. So what we try to set you up with are the basic tools. That's why we talk about enzyme reactions, kinetics, why we talk about the intermolecular forces. Because ultimately all of those are dependent on each of those individual steps. And we think about equilibrium. How does the equilibrium work? Well, each of those is a chemical reaction that is reversible. If we start changing the concentration of one, that's going to change the concentration of 5 other things. Now each of those 5 things affect something else. So it's looking at the overall picture and realizing it is quite complex. So before we actually completely, come on, what I want to do is open up the actual image, whoops, which isn't up there yet. So if you go to the link, that's where I stole the image from. But the nice thing about this image is you can now zoom in. Yeah. So really, really cool. Which is kind of when I saw this image is when I realized, okay, this is interesting, but I don't want to look at it. Because each of those individual steps then can potentially go out and do other things. So what they end up listing, in blue you've got the enzyme names. So what is the enzyme that facilitates the conversion between fumarate to succinate? Okay. Those are the structures of those small metabolites. What things does that enzyme need to function? Well, it apparently needs malonate. It needs FADH2. It needs an acceptor out here to supply your FADH2 to FAD. So you've got all of these intricate little steps that are involved in each and every single one of these reactions. So what we're going to try and do is look at some of these pieces and try and get you to recognize the structural changes and what's happening within those to try and find patterns. Because ultimately, it is, in my opinion, silly to just have you memorize these as raw facts, which is absolutely what you would be expected to do in a biochemistry class. You'd be expected on an exam to draw the Krebs cycle and everything that's involved in it. I don't see any value to that. Okay? So I want you to be aware that that is something that you could theoretically do in a future class, and so you should be kind of thinking about that as far as those processes go. Okay? A lot of it is just going to be looking at reactions and the different contributions within those reactions. I might try and speed that up because I get the impression that most people don't like looking at reactions. I think I tend to be one of the few. So I'll try and speed through some of that, and maybe we'll open up some more time towards the end of the semester where we can talk about some of the DNA and RNA stuff and transcription, translation, replication. I've got a little model thing that I built to help with trying to at least envision what things have to happen on the cellular level to go through and do DNA replication. Okay, which then everything that happens in DNA replication has to happen ultimately for translation or transcription and translation later on, just using different enzymes. Okay, so hopefully that little game will be interesting for everybody when we get to it.
Are we going to be focusing a good deal on that or no?
No. I found it a very boring section for me to talk about because to me what I see as being an instructor is adding value to the material. All that section is a bunch of facts. This is the size of this enzyme. This is the size of this enzyme. This enzyme reacts with this enzyme to make this product.
Well, I meant more of like DNA
So,
replication.
well, even with, and that's, I said enzyme, but the DNA replication, transcription, all of that is ultimately that same kind of thing. Which I guess we could open, we can open to the floor with. Let's go ahead and pause the video. Chapter starts off with chapter 15, looking at free energy changes, sort of. To me, if you're going to talk about free energy changes, you need to see an energy diagram, because that's where free energy is being involved, and they don't really show an energy diagram. Okay? So what they're now referencing is that if we went through and tried to look at a free energy change for any individual reaction, we're going to look at a standard free energy change plus some balance of our products to our reactants. Okay? So this equation should look familiar, because ultimately what this is delta G standard, is it plus RT ln of the equilibrium constant. Okay? So we end up looking at an equilibrium system and saying, what happens if we tweak off of equilibrium? How does that change our resulting delta G? So if we change our product concentrations, our reactant concentrations, what does that do with this? Okay. In 152, or in it's really only 152. If you haven't taken 152, it's fine. They talk about this in more detail, and we mentioned standard state. And there's some things that are typical for standard state. What are these standard states when we're looking at thermodynamics, for those of you that had 152? Your temperature, I think you got to be careful on that one. I think when we're looking at equilibrium, it's actually 298.
Oh, yeah, you're right, it is 298.
298 Kelvin, which is good because that's room temperature. So most biological things were close to room temperature-ish. Officially, we're a little bit higher than that, but that's not too bad. Anything else?
1 PSI or no?
So if we're looking at gases, we would say our gas pressures at our standard state, so this is now in reference to that not there, is that if we were looking at gases, our standard state pressure is 1 atmosphere.
Or 764.
Yeah, I don't even want to bring in units on that. Let's leave it at 1 atmosphere. Okay. So, but
Because they give it to you in torr.
gases, how often are we going to see gases involved in our biological systems?
Every day.
Not as much. What we'll end up seeing will be solutions. That's our primary concern. Okay, so if we're looking at a solution, what's our concentration for standard state? 1 molar. Okay. This becomes problematic. Okay. Why does this become problematic? Most of our reactions in biochem involve the exchange of hydrogen ions through our enzymes. We shuffle hydrogen ions back and forth. Okay? Which means if I'm going to look at my equilibrium or my reaction here, one of those values will be H+. Okay? If I use 1 molar for my H+ at my standard state, what pH am I at? It's a little bit trickier. A 1 molar concentration of H+, that's very acidic. You're looking at a pH of pretty much 0. Okay? A pH of 0 is very, very not good in a biological system. Okay? So instead of looking at our normal standard states for a chemistry reaction, we have to change our standard state conditions. So what ends up happening is we report a different delta G. It's no longer delta G naught. It is delta G naught apostrophe. Okay? Or delta apostrophe naught. Okay? What that apostrophe is in reference to is now a biochemical system or a biological system. And in the biological system, what is our most likely pH? 7. So what ends up happening is that if you look up delta G naughts, those delta G naughts are in reference to a pH of 7 to calculate those standard states. If we then vary off of that standard state by changing the pH, that's then going to show up in that second term. Okay. So it's just something to be aware of. You'll see that little kind of apostrophe show up on your delta G naughts. All that's saying really is that your pH is not based on a molarity of 1. It's based off of a pH of 7, which means the molarity of your H plus is 10 to the minus 7. Okay? So that drastically changes the values that you would end up finding. So if we were going to look at try and predict something happening in a biological system, and we looked up just a delta G naught, that value is going to be drastically different for a biological system because of that apostrophe, because we're looking at that pH change. The other one that shows up, which I don't remember seeing in the textbook, that doesn't mean it's not there, is apparently magnesium ion, Mg+2. If it shows up in your reaction, it has a very particular concentration as well that happens to not be 1 molar. Okay. I don't remember what that concentration is, but it is very particular for your biochemical reaction. So delta G naught is based on pH of 7, okay, or a molarity of 10 to the negative 7, and it's really dependent on your magnesium concentration. Okay. There's a defined magnesium concentration. Okay, so just be aware of those two major changes when we're looking at biochemical systems versus standard systems. Your book brings in some kind of interesting-ish figures. What they're trying to show by this is that we start at ridiculously high energy hydrogen source from the sun. When we then do fission, I never remember which one, hydrogen smashing each other and forming helium, that releases energy. That energy can be absorbed through photosynthesis, okay, and take us to a high energy molecule of glucose. Okay. The high energy glucose can then be converted through metabolism into ATP. We can then use ATP to do all sorts of other metabolic pathways. Okay. Each of those has to loop back down and around. So as we process glucose, to make ATP, we also go through and make CO2 and water. So what we're looking at the top of this curve is all our high energy states. Right. So we got a lot of high energy up top, and we got low energy at the bottom. That says energy. And low energy down here. So our biological systems are designed to kind of capture that energy and control its release somehow. Okay. Each of those steps ends up coming through primary molecules. ATP is one of those big ones as an energy converter. Okay. So in metabolism, we've got catabolism and anabolism, which we've referred to before. In catabolism, we're taking large molecules and we're breaking them down. In anabolism, we're taking small molecules and we're making bigger ones. Okay. So each of those processes is going to do different things. In catabolism, because we're breaking it down into smaller things, we get a release of energy. And you're typically looking at the transferring of electrons to acceptor molecules. Okay. So you're taking electrons from one source and moving them to another source. Ignore the of. I just forgot to delete it. The other big one there is that catabolism is looking at oxidation. Okay. So catabolism and oxidation need to be tied. Anabolism, okay, if we're looking at that process, requires energy. So if I'm going to make bigger things, I need to put energy into it to get it to those bigger states or those bigger molecules. Okay. That's going to involve accepting electrons. Okay. So our smaller molecules need to accept electrons from other molecules to build up bigger and bigger so that we can then connect those linkages through. That's usually using a process of reduction. Okay. So those are your kind of big parallels between those. So if we were going to look at catabolism on an energy diagram versus anabolism, this would be fun. I'm trying to think of some way to remember that catabolism was destruction. Just a cat breaking stuff, right? Look at that. Cat. See? It's kind of funny, right? Catabolism, breaking.
You didn't break the glass.
Wait for it. Okay, so that's enough for fun pictures. Catabolism versus anabolism. We're breaking things down. If we're going to break things down, what's going to happen? Release of energy or absorption of energy? It's a release of energy. So if I'm looking at my reactants, where should I draw my products? They should be lower than it. Because what's happening is I'm now losing that energy out to the system or out to the surroundings. So if we looked at an energy diagram, we're looking at what type of a reaction? Exothermic. Okay. And it's that energy gap that we could potentially use to do something else. Anabolism, if we looked at our reactant energy versus our product, where would our product be? Above it. Higher in energy. That looks kind of horrible, but it's close enough. And the difference in energy there is saying we've got an endothermic reaction, and we have to somehow supply the energy to make that jump. Okay. Well, where can we supply the energy from? How do I get the energy to go from reactants to products here? What's that? Enzymes will change the activation energy, but they aren't going to change the energy between your reactants and products. We need some other type of energy source. So what we end up doing is coupling our catabolism process with our anabolism process. Okay, so we can release energy from one source to supply energy to another source to allow us to stack through. In an organic system, what we end up doing is either changing the energy of our reactants and products by changing the reactants altogether so that it actually reacts more smoothly. Or the other thing we do is we add heat. We add a lot of heat so that our reactant energy gets up high enough that it can clear our activation barrier. Okay. We can't add heat in biochem because what happens if we add heat to living things? Usually die. Okay. So we have to come up with some other way to supply that energy instead of thermal. Okay. Other things that can pop up, this is a slide straight out of my Gen Chem lectures. Okay, if we looked at this overall reaction, we should be able to go through and decide oxidation states for all of the atoms. Okay, yeah, I'm sorry seeing shaking heads on that. Okay, so theoretically you should be able to do that. There's some rules behind all of that. I don't want to get too involved in it because you don't need to go through all of that, but ultimately what you do is look at the oxidation state for all of your atoms in your reactants, oxidation for all of your products, and then we go through to see what changed. So if I go from +5 to 0, what happened? Okay, it had to gain electrons, because electrons are negative. So as I add negative things to a +5, I get closer to 0. Okay, so it gained electrons. If we take a look at the oxygen, nothing happened to it. Stays -2 oxidation state all the way through it. Look at the carbon, went from a +2 to a +4. Okay. To do that, it had to lose electrons. Why lose? Well, our mathematical symbol for loss. Whoa, come on, computer. Whatever, let's just stick with that. Loss is negative, electrons are negative. A negative and a negative is positive. So if we lose electrons, we become more positive. The loss of electrons is most typically defined as oxidation. The gain of electrons is most typically defined as reduction. Okay. You need to know that parallel. In theory, you already do. The more common ones, ways to memorize those is Leo says Gur, or at least that was the most common to me. I've noticed that a lot of people seem to happen to know the other one, which is oil rig, which, I don't know, that's just not inspiring to me. Oxidation is loss of electrons, reduction is gain of electrons. Okay, so I do expect you to be able to recognize those. Okay? I don't expect you to go through and calculate oxidation states for non-organic materials or non-biochemical materials because this isn't an inorganic class, it's a biochemical class. So we'll look at that in a second. The next thing we got to worry about, which we already saw pop up with our sugars, okay, we had reducing sugars. What did our reducing sugar do? It was oxidized. It was not reduced. Okay? So when we talk about oxidation and reduction, you may also hear reference to agents. Okay? So if we have an oxidizing agent. An oxidizing agent caused something else to be oxidized. Okay? It was an agent acting on that other thing. So if it caused something else to be oxidized, it itself had to be reduced. Okay? So if you see that ing, it's pretty much the opposite of what the name sounds like. Right. So be aware of that. That pops up. We saw it already with our reducing sugar, and you may see that pop up again as just kind of general questions. The more important one that I want you to be aware of is what happens to your oxidation states for carbon. Okay, we're looking at biochemistry. Everything is going to resolve around carbon. So that's what we want to be able to pick out, is what happens to our oxidation states through that. Okay? Easiest method is just look at the number of oxygens bound. Okay? So in this first one, how many oxygens are bound? None. In this last one, how many oxygens are bound?
2.
Try again. 4. There's 4 oxygens bound. I know you only see 2 oxygen atoms, but there's 4 bonds. Okay? Which one is more oxidized? The last one. Why? Because it has more oxygens. Okay? So the oxidation name ultimately comes from reacting with oxygen. Okay? That's where oxidation came from. So if you see more oxygen bonds, that molecule is more oxidized. Okay? If we looked at the catabolic process, it was breaking things down. Okay? Looking at a release of energy. The end result, if you remember back to that figure, was what? I know, it was a bit of a stretch.
A lower energy state product.
The lower energy state product, which was?
A release of electrons.
No, what was the molecule?
I didn't see a molecule, I just saw an
CO2 and water.
Oh.
CO2 is our lowest energy structure. Okay? Which is one of the reasons why it's a greenhouse gas. It is so low in energy that it does not react with anything, which means it's virtually impossible for us to actually pull out and stop being a greenhouse gas. Okay? It is an extremely low energy structure. Biological systems like plants have done a great job of changing its energy, okay, by absorbing sunlight and then bombarding that molecule with high energy electrons that now causes it to get up to that higher energy glucose state. Okay? So CO2 is the ultimate end, okay, lowest energy molecule possible. Our carbon with hydrogens is our highest energy source. Okay? Said another way, what's gasoline made out of?
Ethanol.
Try again, it's not ethanol. There's ethanol added to it. It's high what? Hydrocarbons. You see that 89, 93, all those numbers? What are those? It's the percent of octane. Octane is 8 carbons fully saturated with hydrogens. Our fuel source that allows us to move around is a very high energy molecule that is fully reduced. What happens when we try and convert that energy across? We fully oxidize it, or at least we attempt to fully oxidize it so that we can maximize that energy release. Same thing's happening in biological systems. Okay, so we can look at the number of oxygen bonds. As you increase oxidation, you increase your oxidation state. Okay? The next part is you could officially go through and determine the oxidation state for each of your carbon atoms. I might ask that question just because we've mentioned it. The way to figure that out is take your bonds, or take the bonds around your carbon atom in question, and break them heterolytically. So heterolytic means what happens to the electrons in the bond. Sorry, I did that backwards. Crap. Did I? It's not in the textbook. Let's just try it. So if we take a look at this carbon, let's just say it's all hydrogens around it, and we try and determine its oxidation state. If we said we break those heterolytically, the electrons in each of those bonds go back to their parent atom. Okay? No, I think I got this right. So if we break that heterolytically, how many electrons are around that carbon? 4. Now break it homolytically. No, I definitely did that backwards. It's homolytic minus heterolytic. If we break break it in the way according to which atom is more electronegative. Okay? So which atom is more electronegative, carbon or hydrogen? Carbon, which means carbon has how many electrons around it? Then be 8, because it takes all of the electrons from our hydrogens. So I got these two swapped. So we'd have 4 minus 8, which equals -4. Take a look at the oxidation state for our last one now. Okay, and let's do our calculation properly now. It's homolytic minus heterolytic. Let's cross that out. So homolytically means the electrons in those bonds go back to their parent atom. Okay? How many electrons would carbon have in that last structure? How many electrons did carbon bring into the structure? 4. It's its valence count. It's the number of valence electrons. Carbon has 4 electrons. Minus, how many electrons does it have in the heterolytic system? So we take a look at the 2 electrons in one of those bonds. Where do those electrons heterolytically break? So heterolytically means the electrons will go to one atom or the other. They'll go to the oxygen because oxygen is more electronegative. If oxygen is more electronegative, where do all the electrons go? To the oxygen. So how many electrons does carbon have? 0. The oxidation number becomes +4. Oxidation versus reduction, how do I know I actually got this right? Reduction does what to your oxidation number?
Reduction gains. Something gains electrons.
So if it gains electrons, the number gets smaller, it gets negative. So as we go from a +4 to a -4, what did I do to the number? Decreased, also known as reduced. This molecule, our CH4, is the fully reduced form. CO2 is our fully oxidized form. So I did get the formula backwards. It is homolytic minus heterolytic. So homolytic means the electrons go back to the atom that originally brought them, which means you're looking at the valence electrons that each atom brought in. Okay. Heterolytic means the electrons go to whichever atom is more electronegative. What if your atoms have identical electronegativities? Say carbon bound to carbon? Kind of nothing. Each atom gets its original electron. So you end up splitting it homolytically if you have 0 difference in electronegativity. Okay? None of these structures show an example of that. Okay? Because there's no carbon specified up there. Okay? So ultimately what you're responsible for is looking at that difference. Did you oxidize it or did you reduce it? That's going to be your biggest parallel. Okay. If you see an increase in hydrogens, you reduced it. If you see an increase in oxygens, you oxidized it. We'll end there.